Hello to everyone. My name is Alex Galchenko. I am the chief of the Earth Philosophical Society Melodia Vita. In our paper, The Origin of Genetic Code and Translation in the Framework of Current Concepts on the Origin of Life, published in the journal Biochemistry in 2021, my colleagues Lia Kondratieva and Marina Dichkova and I made an attempt to consider and somehow generalize and classify, if you like, main hypotheses for the origin of translation on Earth, which many of our colleagues call the origin of life. The main theory describing biochemical processes in the precellular era is, of course, the RNA world hypothesis. Strictly speaking, the RNA world hypothesis is not one theory, but a quite large number of hypotheses that describe different events but are conceptually united by one idea: the existence of self-replicating molecules that simultaneously perform both informational and catalytic functions. These molecules were, according to the RNA world hypothesis, RNA itself. There is a fairly wide variety of theories for the origin of translation within the RNA world hypothesis. Moreover, the issue of the origin of translation can be divided into several sub-issues. First of all, the issue of the origin of replication and the issue of the origin of the genetic code. Regarding the first, the origin of replication, within the framework of the RNA world hypothesis, the debate is around whether it was catalyzed by ribozymes from the very beginning, or there was a non-enzymatic or rather non-ribozymatic replication of RNA molecules. Orgel and Joyce made enormous contributions to the development of both hypotheses. The theory of non-ribozyme primary replication was greatly developed later thanks to the works of James and Ellington. In the development of ideas about the origin of genetic code, several conceptual ideas arose, which do not always contradict each other. The stereochemical hypothesis was based on the studies by Wöse, who showed that amino acids with a comparable polar requirement are encoded by codons with the same second base back in the 60s of the 20th century. Around the same time, Crick proposed the code expansion hypothesis, the essence of which is that the code was at first dibasic and used a very limited number of amino acids, and then gradually began to become more complex and transformed into the modern genetic code. Wöse also played a huge role in the development of this hypothesis. Crick also proposed another concept, frozen accident theory. In it, he reflects on the fact that as the system became more complex, the pressure of negative selection increased. Accordingly, at some point, the code stopped accumulating changes and froze in its current form. Crick and Wöse also believed that the formation of the observed structure of the standard genetic code occurred randomly and did not involve any optimization mechanisms. However, this doesn't fit well with the fact that the standard code is characterized by quite a high degree of error tolerance. It has been shown, for example, that the probability of achieving a similar level of error minimization by random permutation of codons in the standard genetic code is less than 10 to the minus 6th power. The high resistance of the standard genetic code to translational errors has become main argument in favor of the theory of code formation under the pressure of natural selection. The theory is called the theory of error minimization. In a study of the adaptive landscape of the genetic code, the authors examined the possible evolution of the resistance of genetic code to translational errors and confirmed the hypothesis that the evolution of the standard genetic code could include a stage of its partial optimization in order to minimize the effect of mutations arising during the translation process. In 1975, Wong proposed the metabolic theory. In it, he divided all amino acids into two phases, prebiotic phase 1 and those amino acids that were obtained from their biosynthesis, phase 2 amino acids. Accordingly, phase 2 amino acids inherited the codon of the precursor amino acid. In addition to the region of the genetic code and primary replication, there are several other questions. In particular, how can we explain the current diversity of RNA molecules? We will talk about that in the part 2.